Hello and welcome to BioRules video lessons. Today we are going to be talking about transcription, which in biology is transforming DNA into RNA, deoxyribonucleic acid into ribonucleic acid. The DNA in eukaryotes is placed in the nucleus, so we are going to find it there. Okay, so it's out of reach for the ribosomes that are going to be doing the translation, that are going to be doing the production of proteins. And to produce protein, they need the genetic information. So the DNA, genetic information, in the nucleus has to go to outside of the nucleus in the ribosomes. For that, we use the RNA. So DNA copies the piece of information that needs to be made into one protein, into RNA, and then it goes out of the nucleus to do the RNA. So they get out to the nuclear pores and they are going to be doing the job. So this is how the genetic information from the DNA gets into the ribosomes. Okay, so in this sense, the DNA is going to be the carrier of the information. It contains information, it's the same information as that piece of DNA is going to be having, but it's a short one. It's just one single piece of information for one single protein. One protein, one piece of information, one RNA. Okay, so it's going to be small enough to be able to leave the nucleus through the pores, and is going to be able to get that information into the ribosome. So the letters are going to be sending this information. So RNA, short, single-stranded, and containing information, A, U, C's, and G's. For that, let's remember the comparison between DNA and RNA. Okay, DNA is double helix, RNA is single helix. DNA contains deoxyribose and RNA contains the sugar ribose. The DNA is very, 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 very long. RNA is short. RNA has only the length to have the information to make one protein. Okay, DNA contains a T, C, and G, and the RNA contains a U, C, and G. So DNA contains thymine, RNA contains uracil. Okay, so what's the idea of transcription? Transcription means copying DNA into RNA, which means changing the medium, keeping the language. When you transcribe a video, we get the oral language that's getting out of there and you put in written. We are changing the medium, oral to written, but you're keeping the language, English, Portuguese, whatever language that the video was in. Okay, so this is transcription, keeping the language, nucleic acid, so DNA will still be DNA, but you are changing the medium. So instead of DNA, we are going to have RNA. This is the idea of transcription. And show you what the transcription is going to be about. This is a piece of DNA. Okay, so the transcription will happen by unwinding and doing the RNA synthesis. So we open it and then we copy one of the sides of the DNA into a messenger RNA. And then the DNA gets rewinded, closed, and the pre-messenger RNA is formed like we have there in orange. This is the process of transcription. In a very simple way, in a nutshell, this is it. We open the DNA, we copy one of the sides information into the other side, in the same idea as we did with the replication. If you have an A, you put an U, because RNAs have no T's. If you have a G, you put a C. If you have a C, you put a G. If you have a T in the DNA, you put an A in the RNA. So it's no complementary base pairing. The same ideas of replication, we do that with the RNA. So taking a look here, this is a piece of DNA. That's the three sides. So the 5 side, 3 side, all over there. And I'm going to be opening. And replication happens from a 5 to 3 direction. Transcription also happens from 5 to 3. So if that guy is the new stuff, is going to be copying from 5 to 3. So the direction of replication is going to be this one here. The direction of transcription is going to be also from 5 to 3. So it grows in that way and is growing into the 3 direction. Okay? Then the DNA closes back again. Hey, it's intact. It's the way that was before. It still contains information, but now we have a piece of messenger RNA that also contains that piece of information that can be taken to outside of the nucleus. Okay? So, taking a look at a little bit more detail, the idea is that to start transcription, there is a piece of DNA that's a promoter. It says, here starts a gene, here starts this gene. And then you start the transcription in that point, and you go on all the way until you get to a terminator point, say, here finishes this gene. So we just made a copy of it. This is the idea. Okay. And if you have letters here, this will be a little bit more clear for you. We have, T, A, G, C, whatever. These are the letters. One of the sides are going to be copied. So in this example is the one in the bottom. If I have a T, I put an A. If I have an A, I put a U, G, C, C, G, and so on. I do all the copy. Who is going to be doing this is an enzyme called RNA polymerase that makes the polymer of RNA. And when it's done, the DNA will get out. Okay, let me put it over there. The RNA will get out, the DNA will close back, and you have a messenger RNA done in there. Okay, just a few names for you. The piece of the DNA that's not copied, which in the end is identical to the messenger RNA produced, like here you have an A, here you have an A, here you have a T, here you have an U, okay, because RNAs have U's, but apart from U's and T's, this piece here is identical to the one that was produced. This piece of DNA here is called sense. This is sense because it makes sense. It's the same as the one that was produced. So the other one, the one that's actually being copied, is the antisense. Another name for this is that we call the antisense the template, because it's like a mold. It's the one that's used as a template to make the complementary of the messenger RNA. So if you have a T, you make an A. If you have an A, you have an U. So this is the complementary one, so we call it a template. And the other one, the sense, another name for it is the non-template DNA. Okay, those are the names. Non-template or sense are the ones that are not copied, not the one that's transcribed, and the antisense or template is the ones that are transcribed. Okay, we have to get used to these names because we're going to be using that in exercise during translation. When we finish, we have the pre-messenger RNA. It's pre-messenger RNA because some stuff still can happen in there. Okay, here a diagram showing transcription happening. This stuff is going that direction over there. The new nucleotides are being put in this end. The RNA that was already copied is getting out. The DNA is closing back up over here. So this here is transcription made by RNA polymerase. Another example here, we have it starting at this point over here. The blue is where you have the promoter saying it starts in here, and then it goes on and on and on. It goes making here new stuff. Now the RNA gets out, and it goes close in the back, goes close in the back, and the messenger RNA is done over there until you reach a termination sequence that says here finishes the gene, here finishes the piece of information. Okay. In eukaryotes, things can be a little bit more complicated than that. In prokaryotes, things are simple. Okay, prokaryotes, they have a promoter, they have a terminator, they have information in the middle. You have the transcription, you make messenger RNA, this messenger RNA will do the translation, will produce the protein, that's it. However, in eukaryotes, our DNA is full of pieces that code for protein, like here, or like here. This here have information to make proteins, but also have pieces that do not code for proteins. They are called introns. Introns can be many stuff. It can be rubbish, garbage. It may be fossil genes, genes that are broken and they are left over there. We have them. They can be regulatory pieces of the genes, the DNA. They can be pieces that to make some combinations that will be interesting. I'll show that in a while. Okay, but ultimately introns are pieces that are not going to be giving information to make proteins, but they are transcribed anyway. If it's in between the promoter and the terminator, all the DNA in between those two places, they are transcribed. And now, in eukaryotes, we have to get rid of the introns. So the pre-messenger RNA that have been transcribed entirely from promoter to terminator, now we need to keep the useful information, the exon, and you have to get rid of the useless pieces. By useless, I mean the pieces that are not used for making proteins. So there will be enzymes that work like scissors. They will excise these introns out, they cut and they glue the exons in place. So promoter exon, cut out this intron, and then you have another exon, and then cut out this intron, and then you have another exon, and you have the terminator. Now I have a mature messenger RNA or mRNA mature, and the introns are going to be taken away to be recycled and used again, the nucleotides, to make new messenger RNA or new pre-messenger RNA. And I have here a mature messenger RNA that now can go to outside of the nucleus. Now it can go to the cytoplasm to be translated into protein. Okay, so this is the idea. We made messenger RNA. Okay, another image here showing the splicing, the cut and paste of the introns out of the transcribed messenger RNA. So the idea of the splice is that you may remove the introns, okay? The introns, no coding places of the DNA in the messenger RNA, okay? They may be rubbish, they may be broken genes, but they may also be regulatory. They may help in deciding if a certain gene is going to be expressed or not, okay? Here, an example DNA over here that has been transcribed and then excise the introns. You have the mature messenger RNA that gets out of the nucleus to be made into proteins, to be read into proteins. Another example here, we have the exons in blue. This is in the DNA. We have the pre-messenger RNA done over here. The exons are in the dark red, and we keep the exons, they are glued together, and then we have the mature messenger RNA, just like this. So you have this spliceosome, which is the scissor that cuts out the introns from our RNA game. Okay, so splicing is the cut and paste. And one more detail is that splicing cuts out the rubbish, the garbage, but it may also allow some combinations of exons that may increase the possible proteins that can be built from one single gene. Sometimes you have one piece of information that would code for one protein, but actually, depending on how we can combine the exons, we may make more proteins from one piece of information. Okay? Example, in mammals, there is this protein called tropomyosin, which it makes part of the muscle in mammals. And this gene for tropomyosin contains 11 exons, 11 pieces of information for making protein. However, tropomyosin pre-mRNA may be spliced differently depending on the tissue that is going to be made. Okay? There are 5 different forms that can be made with these 11 exons. Just to give one example, okay? In the striated muscle, in the skeletal muscle that we move around, like our biceps, triceps and stuff, exon number 2 is missing from the messenger RNA, and in the smooth muscle, the ones in your guts, exon number 3 and 10 are not present. So basically, by removing exon number 2 and keeping the rest, we make a protein that's useful in skeletal muscle. By taking out exon 3 and 10, but keeping the rest, including keeping number 2, we make a tropomyosin that's used in smooth muscle. One piece of information, 5 different combinations, 5 different proteins with that one gene. So that means that we can make much more proteins with the number of genes we have than simply one gene, one protein. And this is amazing. Another example, fruit flies, there's a gene called DSCAM that helps guiding the nerve cells to grow into the target. By basically recombining exons in this potentially gene, we can make 38,000 different messenger RNA, making 38,000 different proteins. So basically we can recombine one gene into 38,000 different forms. So this is a lot of recombination. Now from one gene we can make 38 different, slightly different proteins for slightly different functions. Okay? This is the idea of splicing and how the introns can be used for more than, hey, it's just garbage. Okay? Okay, as we're talking about transcription, we should be talking about gene expression, because transcription is what makes the messenger RNA, and once you have the messenger RNA, then you can express a certain gene. If you have the messenger RNA, we can make proteins from that messenger RNA, so we can express that gene. So the promoter says that the RNA is going to be produced, okay, but we might want to produce that protein or not. So for example, here we have a promoter region, and this promoter is only going to be read, read over here by the RNA polymerase if in the promoter region there is a signal saying here, hey, this stuff is open, let's start making RNA to make protein from this. If it's not, then you don't make a protein, then your gene is turned off. We can turn on, we can turn off genes at will. Okay, just to give an example of transcription and gene expression here. In Siamese cats, they all have genes to produce dark pigments in their skin and in the fur. But they only express those genes in the cold parts of the body. If the body part is exposed to a below average body temperature, then they express this gene, they turn on the gene, okay, because they code for the production of this enzyme tyrosinase, and then they produce the pigment. If the body part is warmer than the average, then they don't produce. As a result, the tip of the nose, the ears, the paws, the tails, they are colder than the average and they are dark. This is what gives the characteristic color of Siamese cat, because they only express the gene for color in the cold part of the body. In the warm part of the body, those genes are turned off. Okay? So, this is how we control genes on and off. Okay? So you have this mutant allele CS for Siamese that allows pigment production only in the temperature below average body temperature. This is gene expression, turning on and turning off. Okay? Another example is the bacteria Escherichia coli when they produce lactase, the enzyme that breaks lactose. In the presence of lactase, of lactose, so when the bacteria needs to produce lactase, they unlock this here, they take out this blocker that otherwise would be repressing the production of this enzyme into the bacteria. As a result, this bacteria only produce lactase in the presence of lactose, which is smart. There's no point producing an enzyme to digest a certain food if the food is absent. So if the food is present, they take this out and then the transcription can happen, and hey, we have RNA, so we have the protein. If the sugar, the lactose is absent, then this stuff here gets blocked in place, and then transcription cannot happen. Hey, we're not going to be reading, and this gene is turned off. So bacteria can turn on and off genes, like the gene for the enzyme lactase, just by the presence or the absence of the sugar that is going to be digested by this enzyme. Okay? Another example is when you do methylation or acetylation of nucleotides. So methylation is when you put a methyl over here in this corner. This is a C in the DNA. So let me show you here. I have a C here. Look, we have many C's. Yeah? C's over there. And if you methylate the DNA, then this DNA is turned off. A methylated DNA cannot be read. And if the DNA is acetylated, then it can be read. So basically we can turn on and off genes depending on if you methylate it or if you acetylate it, and you're going to be expressing it or you're not going to be expressing it. So I will show you this here later in class. Here, just to show you an example, in eukaryotes, DNA, they are rolled around these stones. They are rolled around these little proteins. And if the DNA is acetylated, then it allows for these stones to let DNA to be a little bit loose and allows transcription, so it allows expression of the gene. If they are deacetylated or if they are methylated, then they do not allow them to open. This DNA here is going to be locked in place, and therefore it cannot be read. So this here is off, and this here is on, simply by methylating or acetylating a gene. And this is pretty permanent. Like in the cats, if you expose the skin of a cat and you cool it down with ice, of a Siamese cat of course, that part will be exposed to cold. The genes are going to be turned on to produce melanin, and that will be on forever. Later, the cat might not be exposed to cold in that part again when the fur grows and there's no ice, but to still be turned on and produce melanin in that place. So this turning on and off, it may be transient like in the Escherichia coli enzymes, but it may be something permanent. Okay? It's a lot of information. We are going to have time to be discussing this in class. Now do this little quiz here to see if you got the basic information from this class. And please, let's have a very good discussion of this when we meet again in our class. Cheers, bye-bye.